Welcome everyone to the end of the Spring 2020 Science Seminar presentations. Today we have our first presentation in the coronavirus era, which is, that honor goes to Bonnie Smith, so if you'll come over into the camera frame. Bonnie is a native of Brevard, North Carolina. She's a senior biology major and chemistry minor. Bonnie has had a lot of good accomplishments in the last year. She is a fellow for the Young Evangelicals for Climate Action, and she also has been the president of the EPIC Club at Keene, and has initiated a brand new research project here in Bristol that she will be telling you about today. So please welcome Bonnie Smith.
Thank you. So today I'm going to talk about the research I've been working on, ongoing for about a year now, on identifying Lampin flora in Bristol caverns. I do have 3 advisors, some that were from Keene University and some that were from Bristol caverns. So, starting off to do my research, if you're doing your own research, you need funding to do it, and I got my funding through the Lifeford Scholarship that's given through ACA. And I did my research at Bristol Caverns, and that is where I got into it by meeting one of my advisors, Reed Barnett, whose family owns the caverns, and he came to Dr. Allen with the opportunity to do research and identifying what's growing around the lights, because it shouldn't be. So I applied for the Lifeford, and I got it, and I started creating my own research. Boom. So this is a little bit of what the cave looks like. So the cave is a limestone cavern, and it has calcite formations that look pretty cool. They take thousands of years, sometimes millions, depending on their size. There's also a creek that runs all the way through it, and one up the corner you can see it. Hopefully the gutter is not too bad. It's way over here. And calcite, when it is dry and not wet, it is actually very, very sparkly. It looks like glitter. And there is some other wildlife in there. This is a cave salamander. And one there is my favorite. We have a cave dragon. It kind of looks like the mouth of a dragon in the bottom corner right there. So, why do we care about caves? And why do we want to protect them? So other than the calcite formations that are really cool and take a very long time to form, there's also historical important things, like paintings. This one is actually from a cave in Spain. But over time, light or places closer to an entrance that has sunlight, it will grow a little bit of photosynthetic life. So this is closer to an entrance, and that is a liverwort. It's further along on the vascular scale. And this is more algae kind of stuff that I grew and cultured, and would have been around artificial lights. So the fun word that you saw on the first page, Lampin flora, is a German word that if you break down, it's pretty self-explanatory. Lampin as in light, and then flora as in plant. So it just grows in artificial caves. So here's a little bit of Lampin flora that is in Bristol Caverns. This is one of my favorite sections, so you can see Lampin flora in the cave. And hopefully it turns out pretty good on the screen, but there's this really pretty lichen that's like a bright blue color. Its color probably comes from a fungus, but it is growing in symbiosis with other things, and it's a very mixed culture. So this becomes the first problem that you have, that there's lots of different stuff growing all together. So, teeny bit about what Lampin flora can be in there. It's pretty self-explanatory. You bring in light, something learns to live in there. These need light to live, so the light's the main reason it's in there. But it also is in there because it doesn't need as much soil. It lacks the vascular structure as true plants, and has some inorganic substances and uses that rock to break down. So, as it continues to grow, if you see it says blue-green algae, which we now call cyanobacteria, it would be kind of the first stage. It's very, very small, and then as it gets bigger, we go to algae. Then we get into diatoms, which is something I actually cultured. So, the closer to the entrance, as we'll see, you'll see larger things that get closer to true vascular structure, which would just be roots and stems that normal plants have. So This is what it looks like before and after Limp Flora attacks in a cave. As you can tell, this is where the light is shining, so that's why it's able to grow in the cave. And this is a before and after treatment, because you don't want it in there. After a while, it will break down the rock, destroy historical cave paintings. It's kind of a nuisance, a little bit of an invasive thing. So you can really tell, it changes the difference of the calcite formations, and they don't look as pretty. It's also just kind of a nuisance. And I believe this picture was from either France or Spain. Most of the really good pictures that I have were in caves in either of those countries. So, now that I know what the problem is, and we kind of see how it's developed in other caves, a lot in Europe especially, I needed to come up with my research goal on how to get to a plan, because I had to step by step get there. So I started off by, of course, doing environmental samples, and you have to culture them. This one's kind of missing that step. When we do extraction, we get the specific parameters, run PCR to get that little section of DNA that we're going to use to match to check its species, and all the bioinformatics to check that little sequence against all the other sequences of algae so that you find the matching pair to be able to tell you what type of species specifically is there. So using species identification or DNA barcoding, that way is a little bit easier than microscope work. It takes years and years of practice, especially working with microalgae to identify it. So didn't have years and years to learn how to identify it. So a little bit more kind of what we're doing. That was just an overlining. So I won't spend too long, but you have to, a lot of what I had to do in the beginning is a lot of the culturing. This is why this takes over a year and I'm still not quite done. And because COVID-19 is really lovely, it may or may not get finished, because this lovely step right here is actually how they're identifying COVID-19 right now. They're using that, so a lot of the nucleoside base, that kit that I need, is going back for, so when you get to that point, not all of it is finished. So to start off by sampling, you have to have an idea of how you want to evenly sample to represent the cave. A lot of research is done on zoning. So the closer into the cave, starting with C, going out, is the more vascular structure and the more plant, more photosynthetic life you have. You have larger things. This is actually ferns and flowering plants have true vascular structure, and then the rest of them are not quite there. They're getting close. So just kind of zoning it into 3 basic zones. They called it like a day zone, or like the entrance zone, a twilight zone, and then like a dark zone, or a semi-dark zone. A dark zone would have no light. So this is Bristol Caverns, and I'll kind of walk you through where the normal tours are given, so you have an idea before I show you where I picked my sample locations. So the commercial entrance is the entrance that we go in, and then we see these really narrow things. With the lines, those are stairs. So there's 3 levels. You go in, and then down to the up to the bottom level, around, come down here, and this right here with the arrows is actually the running water. There's a creek that flows all the way through the cavern. And then we go up as far into the cave, and then we'll end up turning around and coming back out. This is a section of undeveloped cave right here. Colors just indicate how they're partially done or commercially used. So the places I picked were based off of where they were closer to where we come in, how far in. So they were kind of based off of that zoning that I had read a lot about. So A is very, very close to the entrance, probably about 20 feet at most from the entrance. We have a spot that was underneath the stairs. Oh, then spot B, as it goes further into the cave, the letter increases. Pretty intuitive. One that was partially way in, where you weren't getting that fluctuation of temperature as much from the entrance, but it still had a lot of people coming in and right there, and a lot of people might touch things. Then down below the stairs, and then a spot on the set of stairs that was across from where the creek was. That's as far in as I surveyed. Because of how long it takes to culture, I only did 4 locations. And I would have, when I get further in, I'll probably would have picked a few different spots. So this is what it looks like, the sampling sites up close in person. So this is bees. You can tell there's lots of different, kind of that lightly blue on there. Step A, the humidity is very, very high, and it was actually on the stairs as you enter into the cave. And then C was on the side wall right past when you walk into the stairs. So on top of just taking the idea of the zoning is that the temperature of the light and humidity vary slightly, and that's how you can place it in zones. So I took 3 different samples of each of the 4 locations in about a month period in July, so that they were roughly about the same time of year, had the same amount of people traveling through them, and then I averaged them together to get an idea of what the parameters in the cave are like. To do that, I had used some equipment. It took a little while. It's probably not the most accurate. I had a really tight budget to do this. This little fun thing measures lumens of light. It's a light meter. So it told me the intensity of the light that each section was experiencing. And when we go to the next slide, which is the chart of the conditions, it didn't end up working out in zoning the way I thought. So these are all 4 of the sites, and I put their ranges because there is some very large variations, especially in the humidity. So the humidity varies a lot at site A, way over here, because it's so close to the entrance, and depending on the time of day, usually I would do it in the morning before I started work, but it can just vary a lot, and I think the humidity reader was also just, it takes a while to get situated being underground. I've dilated. But as we can tell, there is no nice, even breakdown of the lights and lumens. It kind of varies. One thing that I really hadn't thought about until you go out and study it in the field is the intensity of light all has to do with how far away the light is from the actual rock. So this one was actually very, very close. It was like maybe this far, but others were a lot further away. So that kind of, the intensity of the light wasn't a very good measure of the light that each site was receiving, but we do have a pretty good progression of temperature that it gets a little bit cooler further in you go, and the humidity does get a little bit more. So the light didn't allow for zoning as much. So now I'm on to the stage of culturing. This is the part that took the longest because algae takes a while to grow. It's not like a C. elegans. It takes a few weeks before you can see it actually on the plate. So to culture, I got most of my resources and where to reference to start with. This is kind of the Bible on algae culturing. And it does say the British Isles, but it is the best book. It has a very good reference for all freshwater algae. So I started off using liquid media, kind of like in these. I'm going to choose 10 media. So algae grows best in a liquid media, and that's great, so you get lots of it going, but you can't get pure cultures because it's all kind of still growing all together. So you have to do isolation streaks like up at the top on my solid media, and the solid media I used was Proteus peptide. These are just general algae medias that would hopefully grow freshwater algae, because I didn't know what species I was dealing with. So I ended up getting 7 different morphotype of algae. These are 3 of them. Hopefully they're decent resolution. Microscope pictures can be a little bit of a challenge. These are live cultures. It's very difficult to sometimes see live cultures on a microscope because the way the light reflects and they move, and you have to have them wet, and if they dry out, you can't see them at all. So getting microscope pictures was a bit of a challenge, probably the hardest thing I dealt with. So we have 3 up here. I have a green. They're kind of really, really clumped. There's probably millions of little that are circular. And you can kind of, wish I had a better one on that. Then this is the red. It has a little bit of filamentous structure. And then my favorite were my pentadiatoms. Hopefully you can see them pretty well, but they're one of the cooler ones. This is the only one that I didn't continue to do DNA extraction on because we could kind of identify it by the symmetry it has. That's what penta refers to. But I think they're really cool. They're pretty important. to get these pure cultures, this is kind of what they end up looking like on a plate. So it's just them by themselves. You don't want other stuff growing in around it. That's what they'll end up looking like. To get to that point, it takes a little while because they first start off looking a little bit like this fun one up here. This is a very old graphic from the 80s. So they actually use like cotton plugs. But you have lots of different things going on, and you start off in a liquid culture, and then you slowly do isolation streak after isolation streak, teasing out till you have just one strain on there. So it can take about 4 to 5 plates, and that's 2 or 3 weeks each plate. So it took a couple months to get those pure cultures. So once I had pure cultures, I was moving on to the step where I needed to extract their DNA. And to do that, I need lots of biomass to be able to do that. So I had to do grid isolations. So this is the green. The green photographs the best, and so you'll see it most. So I just did 16 on there. Each one represents a different sample that I did for the DNA extraction. I had lots of duplicates because you're bound to mess up, and wanted to have good, clean DNA so that I could then amplify it. So I'm on to the DNA extraction part. This part was fun because I had to figure out the protocol. So the protocol I used was a modified protocol for a plant DNA extraction that used CTAB, which stands for my nose, cetyl trimethyl ammonium bromide, and chloroform. But unlike plants, algae does not have vascular structure, so you would break down the kind of grind it up. You're not doing that. Instead, you have to break down and lyse the membranes of the algae. They're very, very tough. To do that, you do a little bit of something different. So we start off using the CTAB, and it works as a surfactant and just gets all soapy and helps break down the outer membrane. And then we put it through 3 cycles of heating it, cooling it, and then shaking it very, very violently with a vortex. And then once you're done with that, it gets washed with a combination of multiple wash cycles with chloroform, isopropyl, and then ethanol, so you get very clean DNA pellets. And you dry it overnight, Then you dry it overnight in your little centrifuge tube, and then you resuspend it in the TE buffer, and then it can go into your freezer and be very, very stable for a while. Once I had all of my DNA extracted, I did the 6, everything but the diatoms. I wanted to see how concentrated and how good the DNA that I was able to extract was. To do this, to test the concentrations, I use a dye, ethydium bromide. So the one that's very, very light right below the arrow of control is just the dye, and this whole row is all of the controls that were known concentrations. So it starts off at about 4.6, 46, and 460 is roughly what it was. So they go up and increasing, and so the other 12 are actually samples that I had to test which one had the closest match to the highest glowing level. This is under a UV. And so this star one, and probably this one's a pretty good match too, but the one with the star is probably the best match, and would probably be selected for the next step in continuing research because it had the highest amount of DNA. So this part took a little while, and I didn't realize this when I first was starting out, kind of how biology major probably know how PCR works, but selecting your primer is, I think, the part that takes the longest with PCR. So I had to pick something that was going to cut the DNA at a specific place that was going to help me identify the sequence. So I needed to pick a primer for an area that was very concerted. So at first I was looking at ribosomal 18S genes, because this is a very important place because you need your ribosome function. So this is one of the primer maps. And I actually ended up using an internal transcribed spacer primer. So I ended up not really selecting all of them on the 18S. The research kind of wasn't, the literature wasn't there as much to find a really good primer that was for general algae use. So depending on which one you pick, so it basically ends up cutting that little section out, and it's usually 30 to 50 base pairs, and then you'll just amplify that. So I picked 3 different primers, and you have to have one to cut each side of it, so it doesn't just have like a very, very long continuous strand, so it's that small piece, so you have a forward and a reverse. So they're not very long. Most of them are about 20, I think the longest is 24 base pairs. And these temperature readings are the annealing temperatures that are important for the PCR. And if you notice, the first primer set has a different annealing temperature. The primers sometimes don't always anneal at the same temperature, so you pick the lower temperature for it to then reattach and replicate and form that new duplicate strands. So as I move on to PCR, this is part that I have not been able to complete yet, but kind of walk through what hopefully I eventually get to finish. So I had already figured out what primers I want. The DNA sample is what was those pretty glowing stuff with the ethylene bromide concentrations. The buffer, and then PCR tubes. The thing that I'm waiting on that COVID-19 has put on back order is the nucleotides. So this is probably a little bit better. So you denature it by heating it up. Most people in biology probably know this. You cool it back down to the annealing temperature, the primer attaches, and then you do this multiple times, 40 times, so you get lots and lots of copies. Won't go over that too much. So this is an example, because I haven't got here yet. But once the PCR is run with all of the primers, I will put them on a gel and run them to see if I have good PCR products, and that if I have enough good clean DNA. Because I can check the concentrations right now before I run PCR, but I don't know how clean it is at the moment. So this, using PCR products, checks how clean the DNA I got was. So now I'm on, theoretically, to the part that is the more exciting, the bioinformatics part, where I'm able to test where each of those little chunks of DNA, what species it comes from. So we don't have the computer or the lab to generate this, but I will send it in to somewhere that had a database, and they would match that strand of about 20 to 30 base pairs, and they would see which one it matches up. If it was a match, it would like exactly line up. This is a chromatograph map, and that's how they check what species it matches with. So at that point, I will know what the species is. So the idea to know the species level is to have a better idea, a more direct treatment plan for the lamprey flora to be able to get rid of it. So there are a few different treatments that people use right now. Most of them are chemical based, which is not the best for a combination of reasons. One, probably the biggest, there's other wildlife in there that can be very sensitive. Salamanders breathe through their skin. It can be very, very bad. Bristol caverns we've recently treated with bleach. A little afraid, I haven't seen salamanders in a bit. Hopefully they're just trying to stay away. A few other things. Peroxide. Peroxide, on the other hand, is probably not as dangerous to wildlife, but it can slowly break down rock a little bit, and that can be a problem. One interesting thing that can be expensive, and that's what deters people from using it, would you change the spectrum of light, or sometimes use a light treatment. So UVC can denature the DNA of the algae and the cyanobacteria, slowly killing it. And you can also change to LED bulbs. So there's a few different treatment options. So there are quite a few things that I would do differently. I was kind of starting off from scratch. I also worked in the caverns, so being in there, I probably worked in there for about a month and a half before I sampled, so that helped me get an idea of where I wanted and kind of just general layout. But I've been in there working for another year, and so there's a few other places that I would like to sample. One, very importantly, that wouldn't hopefully be too difficult to do, would be by the freshwater spring. So it is pretty far into the cavern, as far down underground as you travel, and it does occasionally get light, a little bit from artificial light, but every once in a while it gets sunlight, which is kind of neat because of the way that the light hits it. Another place to survey kind of what the light zone would be, somewhere near the natural cave entrance, or another place even outside of the commercial entrance to see if there's similar algae species able to survive on the outside, because it's very different temperature and humidity inside the cave. So one thing that kind of prevented me from zoning the cave the way I wanted it was light intensity versus how many hours the light was on, and that's what I realized was the big difference. Because the light bulbs, for the most part, there's a few sections that have LEDs, but most of them are incandescent, so they're all about the same amount of lumens that are given out the same amount of light intensity. So testing kind of zoning based off of how long the average of light is on, depending on the section, depends on how long it will be on throughout the day. Because you're going to turn off the further one in, and it's only going to be on for a very short amount of time compared to the one at the big entrance. And one thing that I didn't expect to happen, I put all my cultures in a north-facing window in Whitehall, and they grew there. But a few times throughout the summer and the year, the temperature did fluctuate within that lab. So all of my samples experienced the same temperature fluctuation, but it was not consistent. So somehow trying to control for that, or using an artificial light, because there is an oscillation in the sunlight's intensity throughout the day, unlike an artificial light that's going to be it's just on or off, and it's a steady amount of the same light. So leading into what I would hope in the future people would do with future research, you could do this whole study again, just sampling a couple areas that I think are really good places. Another interesting thing there's not been a ton of research on is quantitative analysis of the algae. So you kind of just looking at what's out there, but once you have an idea, you can see how much, what percentage-wise, how much is cyanobacteria, how much is diatoms, how much is moss, and various other treatment experiments using light, different colored lights, LED lights depending on the color, using EVC to kind of denature it and break it down. And then one really interesting thing, which there's not a ton of research, would be to use more biological treatment. Find something that maybe eats the algae, like a fungus or a bacteria, and try to treat it that way. So I would like to acknowledge Appalachian Association of College, or Appalachian Association, College Association, sorry, for giving me the Ledford, King University for allowing me to do the research, Bristol Caverns, and they are also part of the National Cave Association. I have a few other people like Kelly, who's put up with me asking 400 questions about things, and then Ashley came in for, I used hers Ledford application as a template for mine, and I would have not been able to get it without that. Can I shine any light on your question, or can I shine any light on your questions? So, yeah.
Were the zones, sorry, also behind him, sorry, were the zones in the article that you referenced, was that an unlit cave?
No, it was commercially lit. All of the research that I have is from show cabins, and that's why they're here.
Because it seems like the zones would fluctuate. You know, it sounded like it was based on how far you went in, but
Well,
the amount of lighting that you provide would, you know, modify the zoning sort of.
the zoning is kind of, the graphic that was towards the beginning also talked about zoning, and zoning kind of just has, It's not as much to do with light, it's more to.
Well, and go back even, go
No,
back to
It's
the,
not, sorry, it's way.
so that cave
Yes.
right there. So it sounds like it's based on how far back you go and natural light comes in from the entrance.
So section A will have a lot of natural light, or might be, section B has no natural light, but a lot of traffic and the light's on a lot. And then C is light but low traffic, is
Okay.
kind of how I would determine. And I've seen this in other, this wasn't just like one piece of literature, I've seen it in several other things. And it also, I think the thing about the zoning that's really important is it distinguishes how the vascular structure develops. And because it mirrors evolutionary how plants developed, or photosynthetic life, because this, sometimes algae is closer to protists, but. So you're not gonna, there's a teeny bit of cyanobacteria here, but it's kind of been overtook by all the other things that are in that area. Because over time, it starts off just as a rock surface, and then it breaks it down, and there's more light, and the conditions are better the longer it's been there. It kind of builds off of each other. Sorry if that's confusing. That's one thing I think is important, is how it builds. Yes.
You were talking about maybe doing some experiments that used different colors of light.
Mm-hmm.
Is there any, did you find any literature or any discussion of, with even related types of algae, of what the colors, changing the colors might do? Do you want to have longer wavelengths? Do you want to have shorter wavelengths? Are there specific colors? Is there any sort of rationale or thinking that might guide something like that you ran into? Other than just sort of, pardon the pun, taking the
Yeah.
Edisonian approach
Yeah, and just
and
changing
just
it,
changing the
looking at
light
it,
and
yeah.
seeing what happens.
To start with, before I finished, I'm not 100%, there's quite a few, most everything in the cave, that I got in this moment, will be freshwater green algae. So it'll have some chlorophyll, probably a handful of other, can't remember the names of them off the top of my head. Pigments, these are not as commonly found in vascular plants. And so that's part of the problem, is that we may not know as much about what wavelengths will inhibit its growth, because most of the photosynthetic pigments that we work with plants, because we want to grow food, is chlorophyll. One place in the cave we have kind of tentatively studied, we've used yellow light, it didn't really have much effect.
Do they change bulbs at all to different?
All
do
the
they?
time, constantly.
Yes. Well, have they switched as the technology has switched to
You-
having incandescent more to fluorescent than to LEDs? Have they done that?
Its sections, Bristol Caverns has changed, and it is advised in general with the National Cave Association and just with Lamp and Flora in general to use LED, because it's cooler light, and the temperature will affect it.